Good morning, everyone. We're going to give just a couple of minutes for people to join us. So if you're just joining, we haven't started yet. We've got people coming in, so we're going to give that just a couple of minutes and then we'll get started. Good morning. If you're just joining us, we're about to get started, but we're giving a couple of minutes for the people who are coming into the room. Thank you.
Well, good morning, good afternoon, good evening, everyone. A warm welcome to our Global CDKL5 series, From Science to Hope, Walking the Path Towards Gene Therapy. As today is Rare Disease Day, we're so glad that you've joined us to mark this special occasion. There's really no better way than to get together in hearing the latest updates on Elaaj Bio's progress with gene therapy. I'm Carol Ann, I'm from CDKL5 UK, and I'm joined by my co-hosts and co-producers, Karen from the IFCR and Christina, who is representing the CDKL5 Alliance. And Rodney is also here from the Loulou Foundation. And we're going to hear from Russ Addis of the Loulou Foundation and Elaaj Bio. Together we're here representing our incredible global community. And now I'm going to hand you over to Karen, who's going to tell you a little bit more about what you can expect from today.
Thank you, Carol Ann. As you mentioned, we are excited to be together in celebration of Rare Disease Day, bringing the CDKL5 global community together for this webinar. Today's webinar is the first in a series, and this will provide a high-level overview of the ELJ-101 gene therapy program. This is an exciting and important step forward for the CDKL5 community. We also want to be transparent with everyone about what is still unknown, including details such as clinical trial timing, site locations, and eligibility criteria, both inclusion and exclusion. As we learn more about the program's progress, including regulatory review, our goal is to share what we know today and in the future to set realistic expectations and stay connected and inspired as this work advances along the journey. Now I'm going to pass it off to Rodney for some housekeeping reminders.
Hi, everybody, and thank you, Karen. So just a few housekeeping reminders for today. First, to navigate the Zoom bar options below, you could see that we have enabled translated captions from English to your preferred language. In the chat box, you can see a link to the instructions on how to turn on translated captions from English to your language of choice. If you have questions, please use the Q&A box. Dr. Addis will address pre-submitted questions that were submitted upon registration. Live questions today will be answered as well. The chat box can be used to contact hosts or panelists if you have any specific concerns or needs, and we have temporarily disabled reactions until after the presentation. Finally, the live webinar will be recorded and available by March 7. Everyone who has registered and also through social media, groups, etc., will receive a link. And with that, I would like to thank you all again. And Russ, the stage is yours.
Thank you very much, Rodney, and thank you, everyone. It's a really pleasure to be here. share some slides with you all. All right, should be good to go. Well, thank you again. Really wonderful, wonderful honor to be included by the webinar organizing committee in this first of a series of presentations about gene therapies for CDD. It's great to be here on Rare Disease Day. Happy Rare Disease Day, and it's wonderful that we were able to put this together. So thank you again to Karen, Caroline, and Christina, and Rodney, and everyone else who helped put this together. It's really great. So I'm going to be telling you about the ELJ-101 gene therapy that we're developing at Elaaj Bio. So I thought by way of introduction, I'd give you a bit of a rundown of what we're going to be doing in this webinar. Number one, I want to introduce you to Elaaj Bio. This might be the first time some of you have heard that name. I want to talk about why we're making a gene therapy for CDD, why we think gene therapy in particular is an appropriate therapeutic avenue for us to take. I'm going to talk about how and when we are bringing this gene therapy to the clinic. And finally, we're going to leave plenty of time to try our best to address what questions you may have. So first, what is or who is Elaaj Bio? So we're a wholly owned subsidiary of the Loulou Foundation, a nonprofit that I'm sure the majority of you know, if not everybody. But Elaaj, although you may not have heard the name, it's not brand new. We've actually been around for quite some time. It was founded back in 2018. It was part of a long-standing collaboration with the University of Pennsylvania Gene Therapy Program, working with the laboratory of Dr. Jim Wilson. I'll talk some more about that in the following slides. And our mission is to give CDD patients and families multiple transformative therapeutic options that directly address CDKL5 deficiency. So, moving on to why we are making a gene therapy for CDD. As you probably know, CDD is a very common developmental and epileptic encephalopathy, or DEE. Its prevalence is about 1 in 41,000 births. It presents fairly early in life. Within the first 3 months of birth, seizures can be detected. It has a clinical definition of treatment-resistant epilepsy. And lastly, it is genetic in origin. It's caused by changes in the CDKL5 gene, which is an acronym that stands for the cyclin-dependent kinase-like 5 gene. And that genetic origin is one of the reasons that we think a gene therapy is appropriate, because as we like to say, the CDKL5 gene in this case is both the problem and the solution. It is the problem in that we know that differences and defects in the CDKL5 gene are what are causing the disorder, CDD, but we also believe that it's the solution because this is something that if we can bring back a healthy copy of the CDKL5 gene, we think that we could treat a lot of these underlying problems of the disorder. Now, it's not that there are no treatment options out there. As you all probably know, CDD is fortunate to be one of only 4 epilepsy syndromes with an approved drug. Ztalmy, formerly known as ganaxolone, was approved in the US in 2022 and in Europe in 2023. And it seems that more approvals globally are forthcoming. But this is something that treats the symptom of epilepsy. It does not treat all of the other symptoms of CDD. And as you likely know, CDD is just so much more than only seizures. What you're seeing on the graph here is modified from a voice of the patient report, part of a program that I'll discuss a little bit more. Really asking patient groups, asking caregivers, asking family members, what symptom or ability would you rank as most important for a possible drug treatment today? And in the chart on the right-hand side of your screen, you can see there's a whole constellation of symptoms that people wish could be addressed. And top of the list are things like developmental milestones that you would like to see improved, language abilities, communication, walking that you would like to see improved. Sure, seizures are on there as something that you would like to see reduced, but it's not the only thing. It's not the top of the list. So I think it's important for both the regulatory community and the drug development community to understand there really is still a major unmet need for therapeutics that can address all of CDD's symptoms. So I mentioned the voice of the patient report. That was the product of what was known as a PFDD that occurred in 2019, and that's an acronym for Patient Focused Drug Development Meeting. that was organized by members of the CDKL5 community. And this was an opportunity to speak with regulators and help educate regulators on what exactly it is that patients and caregivers are looking for in a CDD treatment. That was one of several efforts now that have helped to make CDD gene therapy trials possible. And the three organizations, the three studies on this slide here really point to the concept of trial readiness. In the middle is the CANDID study that was organized by the Loulou Foundation, which has global participation. It's still ongoing. There are more than 100 patients enrolled. Some have been there for more than a couple of years now. And the purpose was to generate new knowledge, understanding which clinical measures would be appropriate to use in a gene therapy trial or another trial that's testing a new therapeutic for CDD. We wanted to collect baseline data so that we'd have a reference point, something to compare to as now treatment information becomes available, and really preparing multiple clinical sites around the world to be ready for clinical trials. And the ICCRN or U01 trial is similar in that regard. It's international, over 100 patients, and again, generating new knowledge that can be used by both drug developers, clinicians, and regulators, including the Clinical Severity Assessment or CCSA scale. Together, these trials, these organizations and the work that was put on are the culmination of years of effort from the global CDKL5 community. And I think it's important to take a moment and acknowledge that, all of the years of patient visits and effort put in by all of the families and caregivers here, knowing that these were not trials that were testing a treatment. There was no immediate obvious benefit, but the real benefit comes from these studies now are being used to inform the design of gene therapy trials, including the one I'm going to be talking to you about today. So this is really just a wonderful effort, and it's great to see now this knowledge being put to use. So a little bit of background on gene therapy. This is a diagram that we pulled from the US FDA website. In simple terms, gene therapy is targeting the cause of CDD, the absence of what we call a healthy CDKL5 gene. And so the drug that we're developing, ELJ-101, is an adeno-associated viral vector, or AAV for short. And the idea is that it's taking parts of a virus, sort of a modified virus, to deliver a healthy copy of CDKL5 to the brain of patients. So getting into how we're doing this, wanted to address how and when we're bringing this gene therapy to the clinic. And I apologize, this is a bit of a busy slide, but we thought it was important to show you a little bit more of the process of what's going on in drug development for our CDD gene therapy in particular. It's something that takes many years, and it has a lot of ups and downs along the way. What I can show you is that on the left side of the arrow, you can see their initial work being done building just the first CDKL5 gene therapy vectors to be able to test in research, including in animal models and sometimes in cell models. We then select the clinical candidate to advance what we call IND enabling studies, basically proving that this is something that could be ultimately tested in the clinic. We perform dose finding and safety studies in animal models, both small and large. We have to move to manufacturing of clinical grade AAV. This is material that meets the rigorous standards of regulatory bodies around the world for use in humans. That's a very different standard than what you need to be able to test something in a mouse or in a monkey. The next step is to obtain regulatory approval for the first in-human trial. When we run that trial, the first in-human trial, the focus is on safety. We need to prove first and foremost that the drug is safe in patients. We can then move on to what we call the pivotal trial, also known as the registrational trial. In this case, now we're focusing more on efficacy, really saying, does the drug work in terms of reducing symptoms and improving the patient outcomes? And we can use that to seek approval from regulatory bodies. It will be a great moment when we have approval in the first country, but even then the work will not be done because we want this to be approved in multiple countries so that it is available around the world to the global CDKL5 community. And just highlighting at the bottom of the screen, in parallel to all of this, are the studies that I mentioned previously, CANDID and the ICCRN U01 study, that are still ongoing and still supporting and providing data that can be used in the gene therapy effort that we're developing. So I'd like to tell you a little bit more about the ELJ-101 drug itself. So this is what we call the clinical candidate. This is what the AAV that has been tested now in animal studies and is now moving forward to development in preparation for studies in patients. This is an AAV Hu68 capsid, HU68, which is about 99% similar to the AAV9 capsid that you may have heard of. It yields broad neuronal transduction, and it was developed at the University of Pennsylvania Gene Therapy Program under the leadership of Dr. Jim Wilson. Dr. Wilson is now the CEO of Elaaj Bio, and it's important for everyone to understand that Elaaj Bio has a partnership with Elaaj Bio. We're continuing this collaboration. Dr. Wilson and his team are remaining to be great supporters of our efforts, and they're great helps as we try to take this forward. So now I want to talk about some of the testing that we did of ELJ-101 in mice. Now, what we used are CDKL5 knockout mice. You'll see this referred to on several slides coming ahead with the abbreviation KO for knockout. Scientists have made mice that lack a healthy CDKL5 gene. And so we tend to use the term that the gene is knocked out of their genome, hence the term knockout mice. These knockout mice, it's not fair to say that they have CDD. They're still just mice. These aren't patients. They have behavioral differences from typically developing mice, and they have certain molecular differences in their brains that we can measure and study and learn a lot about the role of CDKL5. But most importantly for drug development is these knockout mice enable us to test our CDD drug candidates so that we can learn how they might work in people. And I'm going to give you an example of the type of study that we've done in these mice. So in a typical study looking for efficacy of ELJ-101 in CDKL5 mice, we dosed newborn mice with a range of dose levels of the ELJ-101 AAV construct. We then waited a couple of weeks and performed a series of behavioral tests. I'm only going to refer to a couple of these, but there are a wide range of testing that can be done. And we can compare the knockout mice that lack CDKL5 to wild-type mice that have the so-called healthy form of the CDKL5 gene. And we can do that in both behavioral testing and in brain analysis. And in both of those cases, what we hope to see is that the knockout mice, when treated with ELJ-101, start to behave more like the wild-type mice. That's the goal of this experiment. So I'm going to tell you a couple of data sets here, show you a couple of data sets from this study. So the first one is looking at a behavioral measure called open field measurement, it's a measure of hyperactivity, which simply put is to say how much do these mice run around? One of the interesting things about the knockout mice for CDKL5 is that they are hyperactive. They run around the box that you see on the screen there more frequently than the wild type mice. And what I'll now do is overlay the treatment so you can see the effect of ELJ-101. So each of the colored lines here, it represents a different dose level of ELJ-101, with group 1 being the lowest dose administered, group 4 being the highest dose administered, and then a couple of dose levels in between. And what you can see is that in each case, as you go higher up in dose, the lines are shifting down closer to that black line of the wild type mice. And this is exactly what you want to see because this is showing that our treatment not only works, the mice are now behaving more like the wild type mice, but it's dose dependent, which tells us that it gives us a really strong indication that it is the drug itself that is causing this effect, because the more drug you administer, the greater the effect. The higher the ELJ-101 dose, the more these CDKL5 knockout mice behave like wild type mice. Looking at another behavioral assay that we use, this is one called hind limb clasping. This is something that is certainly quite specific to studies in rodents. But if you pick up a CDD mouse by its tail, it tends to grab its hind legs together. Its back feet will clasp together. And this is seen in other CNS and neurodevelopmental phenotype mouse models. And so what we're looking at on this first screen here is the black bar is showing the wild type mice, the typically developing mice that have a healthy copy of CDKL5, while the red bar is the CDKL5 knockout mice. And you can see that the red bar is taller because these knockout mice have more of this hind limb clasping behavior. If we then show the result of treatment with ELJ-101, you can see that, again, going from a low dose in group 1 on the left, moving into a higher dose in group 4, that there once again is a dose-dependent effect. And so you can see that the lower doses have a slight impact on reducing this clasping behavior and improving hind limb function, but the higher doses have a more beneficial effect. So again, great to see this treatment is dose-dependent, where the higher ELJ-101 dose causes the CDKL5 knockout mice to behave more like wild-type mice. Lastly, we want to analyze the brains themselves, and we can get a lot of information looking at different protein and molecular signatures in these brains. So there's two graphs on here. The graph on the left is measuring CDKL5 protein in the brains of these treated mice. Now, as you may know, a gene therapy is delivering the gene, which is DNA, and the DNA provides the instructions for then making the CDKL5 protein. The protein is the thing that actually takes action within a cell. And so it is important to show that the protein itself is made after delivering the CDKL5 gene therapy. And that's just what you see. You can see that that the levels of CDKL5 protein on the far right in the wild type, we set that as 1, as sort of a comparator, a standard. You can see that group 5, the knockout mice, have no CDKL5 protein, which we would expect because they're knockouts. And then in groups 1 through 4, you can see, again, a dose-dependent effect. The more drug we used, the higher the amount of CDKL5 protein was observed in these mouse brains. The graph on the right is now showing a different measure. Instead of looking at CDKL5 protein, it's looking at another protein that we know is acted upon by CDKL5. It's a protein called EB2. One of CDKL5's many jobs is to do what's called phosphorylation of EB2. Without getting into all the technical details of that, this is the purpose of this is to show simply that the CDKL5 protein is doing its job. It's doing at least one of its jobs. And again, you can see that in the black box, you would see this is in the wild type mice where essentially this is the standard of EB2 phosphorylation. The red bar in which the knockout mice do not have any treatment, you can see have dramatically reduced EB2 phosphorylation. And then in groups 1 through 4, once again, there's this dose-dependent effect in which higher doses of the ELJ-101 cause increased levels of EB2 phosphorylation. It's worth pointing out it's not perfect, right? It doesn't get to 100%. We're not all the way there, but it is significant improvement and gives us confidence to perform further testing. So these studies in mice, combined with some data that I'm not showing you here, show us that the ELJ-101 gene therapy is effective in mice, and they help us to predict dose levels that will be effective in patients. The next step was to move into larger animals, and this is now talking about ELJ-101 toxicity and safety. So data in mice are very important, but a key step in drug development is to show that a drug is safe in larger animals. And in our case, we used what is non-human primates or monkeys. This is a commonly used approach in gene therapy drug development because the non-human primate brain is very similar in size to the human brain and similar in complexity, certainly more so than is the case for a mouse brain versus a human brain. And without providing all of the details of this study, I can at least share that ELJ-101 passed this test. There were no clinical adverse events. The expression of CDKL5 was detected throughout the non-human primate brain, and there were no significant adverse histopathological findings, which is great news. It means that we can proceed with development of this drug candidate. So to recap a little bit of what I've shown so far and add a little bit more of the steps along the way, the mouse efficacy studies are completed. The GLP, which stands for Good Laboratory Practices, refers to certain internationally recognized standards to make sure that the studies were performed rigorously. The NHP toxicity study is complete. We've had multiple regulatory actions, both with the FDA, MHRA, and others that have given us positive feedback on our program. Most importantly, manufacturing now is underway at a manufacturer in Spain called Virogen, and so far so good. Things are going very well with them. And as I'll discuss on subsequent slides, our clinical trial design has been informed by the results of both the CANDID study and ICCRN. So just to simplify this, perhaps, the mouse studies in the CDKL5 mice answer the question, does it work? Does the drug work? The safety studies in non-human primates address the question, is it safe? And then the manufacturing being done at a scale that can be used for clinical use is really just answering the question, can we make it? And so far, the answer is yes in all three cases. So I'd like to tell you a little bit more about the ELJ-101 first-in-human study that we are planning. The purpose of this study is to determine the safety and tolerability of ELJ-101 while collecting exploratory efficacy data. The goal is to enroll approximately 12 individuals living with CDD, and I should note the trial locations are still to be determined, where our clinical team is still evaluating which sites will be ultimately selected. The dose administration will be one time. This is a one-time dose into the fluids that surrounds the brain via what's called an intracisterna magna injection, or ICM. This again is an approach that has been used by several other gene therapies to deliver AAV to the brain. The duration of this study will be for 1 year with a 4-year extension study to look at long-term follow-up. The assessment frequency during that 1 year will be every 3 months. And many of the things that we're going to be looking at are still yet to be finalized. So I want to be clear that the final protocol has not yet been developed, but these are a list of the endpoints that we expect to be looking at. So as I mentioned, the primary endpoints are going to be the safety and tolerability of ELJ-101 in patients. And then there will be a long list of what we call secondary and or exploratory endpoints. And this is to give us a sense of in what areas might we see improvement in not just reducing seizures, but in cognition, communication, measures of severity, of sleep disturbance. We want to cast a very wide net in this trial because, frankly, no one has done this before. We don't know how patients will respond to having a healthy CDKL5 gene delivered to neurons that don't have one. And so we're not sure exactly what will improve and when, to be honest. And so we want to look at as many things as possible to hope that we can capture the endpoints, the measurements that are going to be most informative and most useful as we think about the next clinical trial in which we're going to be focusing on efficacy and trying to seek regulatory approval. Last note about this trial is that we are anticipating enrollment to open in the first quarter of 2027, so roughly 1 year from now. We do still have a few steps that need to happen to make that occur, but we're happy to say that we're on schedule and excited to be able to offer more information on this as more information becomes available. So if I go back to the slide I showed earlier with our path to a CDD gene therapy, can show you a little bit about where we are. You can see that we've checked several boxes. We've gotten through the preclinical research stages and performing the IND enabling studies. We're now at the stage of manufacturing the AAV that can be used in the trial. Some of this effort has begun, but it's not complete. And so this is where we stand. The next important step by the end of this year is going to be to try to obtain regulatory approval for that first in-human trial that I discussed. And again, we hope that we'll be able to start that trial next year, but even then, there will be a lot of work to come as we ultimately try to reach our goal of approval around the world. As I mentioned, there will be more information coming soon, both as part of this webinar series later this year. I'm hopeful that one of my colleagues will be able to join me as we present more details about the clinical trial that I'm sure all of you are very eager to learn, including where it will be and what exactly the different criteria will be. We've also just launched a website for Elaaj Bio, and it's rather simple at the moment, but I would encourage you to stay tuned, keep track of that. That will be another place that we share information about development on this trial, and also it'll be a place where you can go and revisit this webinar and some of the questions and answers that are provided. Before I turn it over to all the questions, though, I want to tell you one last thing about Elaaj Bio. As I mentioned, our current focus is ELJ-101, this CDKL5 gene therapy, and that is where we're putting all of our resources at the moment. But we do have a vision for the future of Elaaj Bio. We want to see ELJ-101 make it into the clinic and ultimately to approval. But we're working on other candidates as well, including a next generation gene therapy, perhaps using novel capsids, other approaches other than gene replacement, including targeted X reactivation, non-viral methods, things that do not use AAVs. We're looking at gene editing and other approaches. So all of this is to say that while we're tremendously enthusiastic about ELJ-101, we don't view this as the end all, the end of the story. This is something that we need to keep pushing, keep trying to optimize, keep trying to get better and better treatment options for patients and families with CDD. With that, I'd like to thank a lot of folks. Number one, the families and carers and caregivers with CDD, the Global CDKL5 Community, including all of the family, patient advocacy groups, particularly the Loulou Foundation, CDKL5 UK, IFCR, and the CDKL5 Alliance. Really wanted to give a great thank you to all of the participants and family members in both the CANDID study and the ICCRN. As I mentioned earlier, this is really what is enabling this gene therapy trial to move forward. And finally, I want to thank our many partners and collaborators, including Gemma Bio and Dr. Wilson's group, Elpida Therapeutics, that has been a great help to us, as well as Mazi Therapeutics, who have been wonderful supporters of our efforts. And with that, I'm going to turn it over to some questions. My understanding is that there were quite a few submitted in advance. We can start there, but then I've been told that there are quite a few questions coming in the chat boxes, and so I'll stick around and we'll be happy to try to address as many as we possibly can. Just to highlight here that we thought it was wonderful that we received so many questions from around the world and in so many languages, and we'll address these as we move forward, but just wanted to say thank you for the engagement of the community around the world. And I think for now, Rodney, you can interrupt me if I've got this wrong, but I'm going to go ahead and do my best to answer the first series of questions that were submitted in advance, and then I'll turn it over to the moderators to read some additional questions. And I certainly understand that questions submitted in advance is a challenging thing, because if you don't know what I'm going to say, it's hard to know exactly what are the appropriate questions, right? So things like, when will the gene therapy be available? As I said, we think that the first trial will open next year, but even then, that's available to a relatively small number of patients. There will be a subsequent trial following that, hopefully within the next 2 to 4 years, and then approval in first one and then other countries. As I said, the clinical trials will begin in 2027. I think we addressed the trial duration. It's going to be a 1-year trial with a 4-year long-term follow-up. We have not yet committed to trial sites in the USA or elsewhere, so I can't speak to that. We're still evaluating the sites and the principal investigators who will run these trials. And I think we addressed the timeline for the trials. Details on eligibility is something that's still being finalized, and including the criteria for inclusion and exclusion. When will we know them? That will be later this year, and we'll be happy to share that in a subsequent webinar. And again, on the website, we'd like to provide that information. As of now, do not have any limitations on specific CDKL5 mutations. We are intending to develop this therapy as something that will be available to all patients, regardless of mutation or age or gender, so that it does include the deletions. With regard to participation of children at specific ages, I can say that, during drug development, there are sometimes reasons to restrict the population at certain early trials, but the goal in the long term is to make something that can be usable by patients of all ages. The question about, do you believe that older adults with CDD will benefit from the outcome of your efforts? The truth is we don't know, but I can say that we have some hope. As I mentioned earlier, this is something that has never been done. No one has ever given older adults or younger adults with CDD a new copy of CDKL5, and so we won't really know until we try. However, we do have some reasons to believe that this could be beneficial. Number one, we know that CDD is not a degenerative disease in that neurons are not dying over the course of the patient's life, as is the case in some other severe neurological disorders. And so the neurons are still there. They're just not functioning the way they might if they had a healthy CDKL5 gene. So that gives us some hope that there could be benefit even for older adults with CDD. And then the second thing I would share is that in mouse studies with the CDKL5 knockout mice, there have been some very interesting experiments done in which older mice, adult mice that lacked CDKL5 had their CDKL5 gene turned back on. And this is a sort of a genetic trick that we can really only do in mice. But those mice did show benefit. Again, this is heavily caveated. I don't want to read too much from mouse data and say that it's going to predict what will happen in patients. All I can say is that it does give us hope, and it does give us a reason to think that it is at least worth trying to treat patients of all ages. The question about when will this begin human trials, and specifically for boys, the human trials will begin next year. And again, we have not yet finalized the eligibility criteria, whether it's boys and girls or both. So that'll be detail that we can share later this year. There likely will be some, perhaps not limitations on current medications for children, but this will need to be tracked, and all of this will become clear as we finalize the final protocol of the trial. Questions about locations. So, for example, given patients' travel challenges, would we consider a collaborative framework to run the studies in multiple countries, including Turkey, with regular updates? Or is it possible to participate from Germany? As I said, our goal is to make this accessible to patients around the world. Some of that is going to be dictated by the trial sites themselves, and that is still being finalized. And there is also opportunity for patients to travel as well. So I'm sorry I can't give a more detailed answer to that. I know it's a bit unsatisfying. All I can say is that more information will be available later this year. Clinical trial, as I mentioned, will have an initial duration of 1 year with a 4-year follow-up to look at both safety and efficacy endpoints. How could my daughter try ELJ-101? We're in Kuwait. Again, once we establish the trial sites, I think it will become a bit more evident about how folks might be able to travel to receive to participate in the first trial. But once again, there will be other trials beyond that, and certainly we hope approvals. If the trial will go to phase 3, will it be available for Italians and European patients? My hope is yes, but again, that will require us to understand, in a subsequent trial, what are all of the sites. The hope here is that this will be available to patients around the world. And as I mentioned, the study locations are still being determined. When can we expect FDA approval from the first human trials? And how can we register from South Africa? The FDA approval process is something that, to be honest, we're not certain that we're focused only on FDA approval. We're looking at multiple regulatory bodies around the world. So that although we certainly would like US approval and FDA approval, I don't know that that is going to be the first that we seek to achieve. But that would then allow immediate access to patients in the United States or patients who can travel to the United States. We're going to also, in parallel, be seeking approval in other jurisdictions so that hopefully it will be available around the world. What are the results of gene therapy in animal models? I think we discussed that in the slides today. Certainly in the CDKL5 knockout mice, we are happy to see dose-dependent improvement of various behaviors associated with CDKL5 deficiency. For patients who have already developed irreversible developmental and cognitive impairments, can your gene therapy theoretically cure these issues in the later stages of childhood? This goes back to the comments I made on a previous question. In fairness, we don't know, but it's also not clear to us that we should use terms like irreversible impairments. We don't know yet if those impairments are reversible, and that's one of the main things that we're aiming to learn from this gene therapy trial. What are the biggest risks researchers are watching for with this gene therapy? I think with regard to the actual clinical trial, as I mentioned, the first trial is going to be focused on safety. There have been other gene therapy trials where you've seen severe adverse events, in some cases even patient deaths. So the safety of the trial of the therapy has to be paramount. That has to be the first thing that we focus on. But there are other risks along the way, just in development, even in our manufacturing that's happening now. A lot of gene therapies get tripped up and slowed down by delays in manufacturing. There could be delays in the regulatory approval process. There are all of these things that we're mindful of and trying to mitigate those risks to be able to keep things moving forward. There's a question about what percentage of overall CNS neurons was achieved in primate studies of the Elaaj gene candidate. I like this question because it happens to be one of the first ones that I asked when trying to look at the non-human primate studies. And what I can say is that the team in Dr. Wilson's lab that ran this study did not measure the overall percentage. They did look at expression of the CDKL5 gene and saw it throughout the brain. But I do agree that it would be very interesting to know what is the exact percentage and perhaps be able to compare that to other gene therapies. That's the end of the pre-submitted questions. I'm going to stop there and I'm going to stop screening, and hopefully we can take some more questions from the group.